From here, we will see the rest of the eyeball. So the two cavities we spoke of earlier, the larger will be the posterior cavity containing the vitreous body. The smaller will be the anterior cavity containing aqueous humor. These liquids help maintain shape of the eye, creating intraocular pressure. Okay, and we briefly discussed how things are divided. The anterior cavity has an anterior and posterior chamber divided by the iris. Anterior chamber between the iris and cornea, filled with aqueous humor. Aqueous is clear and watery. Aqua, aqueous. The posterior chamber will lie behind the iris and in front of the lens, filled with aqueous humor as well. Posterior chamber, filled with vitreous humor. It is transparent but more gelatinous in substance, also known as vitreous humor. So this is the pathway of light we spoke of earlier. Here we see that cornea. Light passes through into the anterior chamber, past the iris through the pupil, posterior chamber, through the lens, through the vitreous or posterior cavity to the retina. But here we can see where aqueous humor is produced in that ciliary body, and it moves through the posterior chamber, through the pupil, then into the anterior chamber, and then is reabsorbed in the sclerovenous sinuses. So the lens is very important when it comes to refracting light or bending light. This is to focus that light on the fovea centralis of the retina. So that bending is happening at the cornea as well as at the lens. Okay, focusing light on the retina, and then the ciliary body is controlling the shape of the lens. So refracting light, if you've ever looked into water and you go to reach for, say, a rock, you'll notice that when you do, it seems your arm bends in a slightly different direction than you initially thought. This is refraction of light. Okay? So when light is focused on the retina, the image is actually inverted and right to left reversed due to this refraction. Luckily, our brain will help correct this image, and this takes place in the occipital lobe. The lens will accommodate to properly focus that object on the retina. Image is projected to that central fovea or the fovea centralis, where we have our most acute vision. This is a picture of how an image will pass through the cornea and the lens and be reversed and inverted. So normal vision is going to be emmetropic. An emmetropic eye or emmetropic vision will refract light correctly onto the retina. We all know somebody who happens to be nearsighted or farsighted. So nearsightedness will be myopia. So that eyeball is usually larger than what it should be. So this will move the retina farther than what it should be for light to converge. So it will converge before the retina or in front of the retina. So these people can see close objects sharply, but distant objects become blurry to them. For correction, we have concave lenses. So here is a view of that. The eyeball becomes more elongated than what it should be. The reverse of this is hyperopia or farsightedness. So this is when the eye is shorter than what it should be. And so the convergence of light is behind the retina. So distant objects are clear, close objects are not. We use a convex lens to correct these. And astigmatism is where the cornea or the lens, or even both, have irregular curves. This is going to cause blurred or distorted vision. Instead of the eye having an abnormal shape, we have the cornea or the lens causing the problem. But here's hyperopia. So rods and cones, again, are photoreceptors, and we convert light into neural impulses. So these photoreceptors get their names from their appearance, rods and cones. Okay, so they're going to contain photopigments, which will absorb the light and lead to the production of the receptor potential. So rods will only contain rhodopsin, and cones will contain 3 different photopigments. This allows us to see red, green, and blue. So photopigments respond to light in a cyclical process we'll see here in a bit. So the neural pathway, light hits the retina. We detect light through rods and cones. Those rods and cones move that energy through the retina to the ganglion cells. Those ganglion cell axons move towards the center of the retina at the optic disc, or towards the optic disc. This will create the optic nerve. From the optic nerve, we will move to the optic chiasm, which we'll see in a second. From there, we split and create the optic tract. And then we move through the lateral geniculate nucleus of the thalamus, and then the optic radiations, allowing information to arrive in the visual areas of the occipital lobes. So here's this optic nerves would be seen here. This is the optic chiasm. You will see some crossover as well into the optic tract, through the thalamus, optic radiations into the occipital lobe. Okay, so vision, our vision is binocular. By this, we see 2 visual fields that cross each other. So we have a medial and lateral, or temporal and nasal fields. So the right half of each visual field travels to the left side of the brain. Visual information from the left half of the visual fields travel to the right side of the brain. What that means is anything that we see on the left side of our body will contact the right side of our retina, and those signals will move to the right occipital lobe. So anything on the right side of our body will cross over, contact the left side of the retina. Those will either cross or stay on the left and move to the left occipital lobe. So over here, if we contact the left side, we stay on the left side. Over here, if we contact the left side, we stay on the left side as well. So that means we must cross over at the optic chiasm. Just the opposite. Since we're in the right eye, anything hitting the right side of the retina will stay on the right side, entering the right occipital lobe. And that is it for vision.